Bismillahirrahmanirrahim. Good morning, ladies and gentlemen. I am Endah Larasati from University of Indonesia. Today, I would like to present my research. The title is Confirmation DNA Damage through In Vitro Study of Formation DNA Adduct 8-hydroxy-2-deoxyguanosine as Biomarker caused by acrylamide exposure. The first is Introduction. The reason why this research is being important to do is the sources of acrylamide are very convenient in life. Because the sources of acrylamide are very convenient to find in life. Acrylamide was there in potato chips, coffee, and cigarette smoking. So, individuals have many opportunities to be exposed to acrylamide. Acrylamide is classified by International Agency for Research on Cancer in Group 2A. It means acrylamide is a chemical that has effect probably carcinogenic in human. Acrylamide is phenylic compound that has a low molecular weight and body easily absorbs through skin, inhalation, and gastrointestinal tract. Metabolism of acrylamide in human body generates reactive oxygen species as byproduct. One of the most reactive types of reactive oxygen species is hydroxy radical. It can interact with DNA molecules such as 2-deoxyguanosine at C8 in 2-deoxyguanosine lead to formation of 8-hydroxy-2-deoxyguanosine and induce DNA damage, as you can see in figure 1 and 2. 8-hydroxy-2-deoxyguanosine can be repaired and excreted from the body, so 8-hydroxy-2-deoxyguanosine is considered as a marker to predict the occurrence of oxidative DNA damage. In vitro study of 8-hydroxy-2-deoxyguanosine caused by acrylamide exposure to 2-deoxyguanosine as one of DNA-based lags. So this research is conducted to identify of the formation of 8-hydroxy-2-deoxyguanosine as biomarker DNA damage caused by acrylamide to 2-deoxyguanosine. Okay, next turn to experimental methods. In this study, I conducted 3 steps. The first, I'm going to look for optimum condition from high performance liquid chromatography as the instrument to detect 8-hydroxy-2-deoxyguanosine. Include look for the composition of mobile phase and flow rate. And the second one, I will do in vitro study of 2-deoxyguanosine and hydrogen peroxide. And the last, I will do in vitro study of 2-deoxyguanosine and acrylamide and hydrogen peroxide. So, for in vitro study of 2-deoxyguanosine and hydrogen peroxide, for the first step, I will take 100 microliter 2-deoxyguanosine 10 ppm in 0.1 molar buffer phosphate solution with various pH 7.4 and 8.4. And then the next, add 100 microliter hydrogen peroxide 100 ppm. And then incubate in shaker bed for 24 hours at 37 Celsius. And the result for this incubation will detect in high performance liquid chromatography. For in vitro study of 2-deoxyguanosine, acrylamide, and hydrogen peroxide, for the first step, take 100 microliter to deoxyguanosine 10 ppm in 0.1 molar buffer phosphate solution with various pH 7.4 and 8.4. Then add 100 microliter acrylamide 100 ppm and add 100 microliter hydrogen peroxide 100 ppm. And then for the next step, incubate in shaker bath for 24 hours at 37 Celsius. And this result will be detected in high performance liquid chromatography. Okay, for the next slide, results and discussion. For the results and discussion that I get, for the first is optimum condition that obtained to detect 8-hydroxy-2-deoxyguanosine is using composition of mobile phase 85% buffer solution and 15% methanol with flow rate of 1.2 milliliter minutes. And from the linearity, I obtained the value of a regression equation y equals 0.5097x plus 0.0855. And for the coefficient correlation, I get 0.9985. And for limit of detection and limit of quantification high performance liquid chromatography, I obtained the value for LOD is 6.108 ppb, and for limit of quantification I obtained 20.361 ppb. Okay, before I inject the sample, I inject a standard solution of 8-hydroxy-2-deoxyguanosine 100 ppb. So from this figure 1, we know that 8-hydroxy-2-deoxyguanosine was detected in 5.2 8 minutes. Then from the sample, that incubation 2-deoxyguanosine, hydrogen peroxide, and acrylamide in pH 7.4 for 24 hours and 37 Celsius get a retention time of 8-hydroxy-2-deoxyguanosine is 5.35 minutes. So this retention time is close to retention time for a standard solution of 8-hydroxy-2-deoxyguanosine. So I guess that this peak is 8-hydroxy-2-deoxyguanosine. And for the next figure, in figure 3, describe that in incubation 2-deoxyguanosine and hydrogen peroxide at pH 7.4 was yielded as much as 1.891 ppm. And then at the pH 8.4, incubation 2-deoxyguanosine and hydrogen peroxide was yielded 2.151 ppm 8-hydroxy-2-deoxyguanosine. But when added acrylamide, when acrylamide was added to that same condition, in which at pH 7.4 and 8.4, From this incubation was yielded 1.992 ppm, and this condition and with the same condition at pH 8.4 was yielded 2.617 ppm. So according to this data, acrylamide increased formation of 8-hydroxy-2-deoxyguanosine. Then, according to the result, incubation of 2-deoxyguanosine and hydrogen peroxide generated 8-hydroxy-2-deoxyguanosine due to contribution of hydrogen peroxide, which produced hydroxyl radicals. The equation for this reaction there's in equation 1 and 2 like this. So, and the formation 8-hydroxy-2-deoxyguanosine by hydroxy radical showed by figure 4. Then, based on literature, exposure of acrylamide by in vitro study induced the formation of 8-hydroxy-2-deoxyguanosine, and the results of this study represented that acrylamide contributes to the formation of 8-hydroxy-2-deoxyguanosine. It was evidenced by an increase in the concentration of 8-hydroxy-2-deoxyguanosine produced when 2-deoxyguanosine was incubated with hydrogen peroxide and acrylamide at pH 7.4 and 8.4 at 37 Celsius for 24 hours. In this study revealed that the highest concentration of 8-hydroxy-2-deoxyguanosine was at pH 8.4, as much as 2.617 ppm, as you can see in figure 3. According to the results, increasing of formation 8-hydroxy-2-deoxyguanosine caused by acrylamide is in the higher pH. It represented that in the higher pH, acrylamide contributes to induce 8-hydroxy-2-deoxyguanosine formation more than pH 7.4. In conclusion, the exposure of acrylamide and hydrogen peroxide to DNA base, typically 2-deoxyguanosine, formed DNA Adduct 8-hydroxy-2-deoxyguanosine. It's probably that acrylamide generated reactive oxygen species to form 8-hydroxy-2-deoxyguanosine. Increasing of level 8-hydroxy-2-deoxyguanosine was obtained in the higher pH due to elevated rate of decomposition, hydrogen peroxide, and contribution of acrylamide that produced the increasing of 8-hydroxy-2-deoxyguanosine formation. That's all my present. Thank you.